Okay, everyone, welcome back. So today we're going to cover the central dogma of molecular biology, the part on translation, which you're going to take notes on page 32 and 33. Please make sure you Cornell these notes. Pause after this slide in order to create the Cornell format. Okay, so today's EQ is how does a cell use our RNA or ribosomal RNA to synthesize protein? Um, so we're going to go over how a cell actually uses the information from the DNA that has been taken out by the mRNA and turns that into a protein. So what we know is that so far what we know is that we use DNA to create the RNA called mRNA or messenger RNA, and then that is used then to create the protein. So now we're going to go into the second part of protein synthesis called translation. So we're going to quickly go over what this word means, and and then we're going to go over the process of making the protein using the information that the DNA has sent out through our mRNA. So, transcription is making a copy. So, why do we need to translate RNA? Well, RNA is made out of these things called nucleotides, such as adenine, uracil, guanine, cytosine. And protein is made out of amino acids like proline, methionine, serine, valine, leucine, and methionine again. So we basically, they're two different languages or kinds of molecules. So we've got nucleic acids in the that makes up the RNA, and then we've got proteins or amino acids that make up protein. So we're talking two different very different types of molecules. Translation. So to translate something means to write something in a different language. So here at the bottom, we've got, hi, what do you want to eat? Translation into Spanish would be, hola, ¿qué quieres comer? So we're looking at a translation. We're translating one thing into another. And that's what we need to do to the information from the nucleus that's coming out from in the form of mRNA. mRNA needs, the information needs to be translated into something your body can actually use. So your body can't really use our RNA or messenger RNA. It's actually made out of protein. So that information has to be turned into a protein that your body can use. Okay, so you're going to be seeing a graphic, or you already saw a graphic of the mRNA there in red is going to leave the nucleus and meet up with the ribosome, and that's it. The first step of translation is that once the RNA is made, which is called transcription, it leaves the nucleus. The mRNA then leaves the nucleus and meets up with a ribosome. Okay, so step two of the process of translation is that after the mRNA leaves the nucleus, the ribosome attaches to the mRNA and it starts reading it and it starts looking for what is called a start codon. A codon is made up of three basically nucleotides, and all ribosomes basically are turned on by the sequence AUG. So a ribosome starts doing its job when it gets to the mRNA sequence AUG. And so we found this out and we called that the start codon. So AUG is known as the start codon for ribosomes, and that basically turns on the ribosome. So the ribosome will start looking for that particular code in the mRNA after it leaves the nucleus. Okay. So step number three. After that, the ribosome, now that it's met up with the start codon, it starts reading the mRNA and it starts reading it in triplets called codons. I already told you last slide that it reads in threes, in sets of threes. So a ribosome only reads in sets of threes, and we call those codons. So then it starts reading it. So that's step three. So, step four is that after the ribosome starts reading the mRNA, uh, it moves. one codon at a time or three nucleotides at a time. So the next reading frame would be CUU after AUG right here, CUU, and basically a molecule of tRNA is matched up to that. So you have tRNA that's one type of tRNA that's matches up. So you'll have a tRNA that says, "Hey, that's what I'm carrying. CUU is my thing." So it brings in the amino acid leucine in this case. So the tRNA molecule will swoop in and basically drop off the proper amino acid based on the code CUU. And that's step 4. Okay, step 5. The amino acids connect to form a long chain called a protein. Okay, so here we're going to start where we left off in step 4. We have the tRNA that was carrying leucine that recognized CUU. And here, the tRNA will drop off the leucine hand it off to the ribosome. The ribosome takes the amino acid leucine, and the tRNA basically leaves and goes to find this particular tRNA will go to find another leucine to attach to itself. But it drops off that leucine, hands it off to the ribosome. Okay, so continuing with step 5, the next thing that happens is that the ribosome will scoot over to the next reading frame, which in this case is CCC. So the ribosome will kind of scoot over and land on the next reading frame, which is the next codon, which is CCC. And then the tRNA that recognizes CCC says, "Hey, that's my thing. I'm carrying that amino acid that matches up with CCC." Another tRNA will come in and say, "Hey, that's mine," and bring in the correct amino acid, which is in this case proline. So the tRNA carrying proline will come in and match it up, and so then the tRNA will basically drop off the proline to the ribosome. Okay, so here we're still on step 5. Okay, so in this slide here, we see now that the tRNA dropped off the proline and left, and now the ribosome continues on to the next codon or reading frame, which is AGU. And the ribosome will scoot over 3 more. So now it's AGU, and the tRNA that matches up with AGU will bring in the correct amino acid that it's carrying. In this case, it's glycine. So then that tRNA will come in, drop off the glycine, and hand off that glycine, that amino acid, to the ribosome, and then the ribosome will connect it to the growing chain that right now has leucine and proline, and the third amino acid is going to be lysine. So now what happens is that the ribosome, like before, moves on to the next codon, which in this case is GGU. The tRNA that matches up with GGU will bring in the amino acid. In this case, it is valine. So the tRNA brings in the valine and drops off the valine to the ribosome, and the valine then gets attached to the glycine, forming the chain. Finally, step 6. Step 6, the ribosome continues on reading, and it reads it basically starts translating the next codon, which is UAA. Well, UAA is one of the 3 kinds of stop codons. And stop codons are UAA, UAG, and UGA. And what these do is that they tell the ribosome that that's the end of the sequence of amino acids. There's no more amino acids here. So what it will do, it basically disengages from the mRNA. It comes off the mRNA. And releases the amino acid chain. So here you've got the ribosome on the stop codon UAA, and it is now releasing the chain. and it is going to disengage from the ribosome from the mRNA and leave. So once it reaches a stop codon, you can see here the ribosome has left. The mRNA has not been changed. It's still there. It can be another ribosome can come in and attach to it and start reading it again. But more importantly, you've got your leucine, your proline, your glycine, your valine all in a chain, which we call amino acids in a chain. We have a name for that, and that would be protein. So we now have a protein or an amino acid chain. So, this particular picture was obtained from a higher level book, but basically here you get the picture. So, here we've got DNA. Transcription gives you RNA, more accurately mRNA. We get that mRNA and we use it in the process of translation and create a protein from that. So here, you have to remember that the mRNA is broken up into triplets called codons because the ribosome reads in frames of 3, so a codon would be 3, and those code for the tRNA brings in particular amino acids. The ribosome will attach them into a chain, and you get yourself basically a protein, which is what our body is. So, as a kind of summary quiz, here are 4 questions that you're going to be answering, and they are going to appear. They're going to pop up, so make sure you answer these accurately. And good luck. This is basically the chart used that we figured out, basically, how to read the codons. So we figured out how to decode the mRNA. So we know how to read mRNA, believe it or not. And this chart right here gives you the amino acids that are appropriate for certain combinations. And I'm going to show you guys how to read this so you guys can use it to decipher the code. So, the first thing you have to do if you want to get CUU, okay? So the first base is C. So you look on the left side of the screen, you find, you look up and down. The first base is not U, it's C, right? It's not A, it's not G. So you're going to look at that right there in the yellow. And so C. And then you're gonna go, okay, well, we got C. And now the next one is U. So you look across the top, the second base. So the second one is U. So you find U across the top, and it's right there. It's in pink. So the U, okay? And then the third one is another U. So you look along the side, and you have to kind of look at the first one. It tells you where to go. So you look at the first. It's kind of like the second row, really like the second row. So you kind of find U because that's the last base. The third base is U, and you kind of figure out where they come together, and that's how you figure out where they come together. So if you did the part correctly earlier, you know that the amino acid is leucine that corresponds with CUU. So CUU corresponds with the amino acid leucine, and this is how you find it. You found the C on the side, you found the U up top, and then on the right hand side you find the U, and that tells you it's the first little line right there, and that gives you leucine. Okay, so there's this chart right here that is often used. The next part, there's 2 types of charts. This is one kind of chart, so if you know how to read it, that's great. But there's also a second type of chart that I'm going to teach you how to read also. Okay, so this is another type of chart that's also used. And this one, it's basically the first codon would have to be on the in the center. So if we're trying to find CUU here, we're going to go to on the right side, you find C in the middle. And so there's C, and then on the second ring around, towards from the inside out, you find U, and there you see it to the right. So it's CU, and then you find the next U, which is on top there on the third ring, and that gives you leucine. So we've got CUU right there, and that gives you leucine again. So both this chart and the other chart give you exactly the same thing. They're just in different formats. So here we've got the 5 steps in an abbreviated form. Feel free to use this as part of your summary. because it nicely summarizes exactly what happens every step of the way of translation. For your final task, you will be coming up with the amino acid using the charts. So you're going to have to rewind a little bit to where the charts are, and either the round chart or the box chart, and figure out the amino acids that go with these. You can also open a book. I believe it's on page 303, that you have the round one, and you can use that. It's in your book, and you can use that to try to decipher the chart. Um, and try to figure out which amino acids go with UAC, GCA, UUA, AUG, CA, GAU, GCC, AUU, and AAG. So good luck, and we'll talk again soon. Bye.